All right.
Here we go.
Can you guys hear us on Zoom? Give me a thumbs up.
Let's see if we can create a feedback loop. Hello, hello, hello. Yeah, it's coming through.
Awesome.
Because my Zoom was muted, so it was coming from here.
Okay, great.
And
I
then
can double check if I just pulling up the Zoom call.
last step is going to be to make sure I can share.
Austin, can you hear us?
All right, I just tried to share. And it says that host disabled participant screen share.
I got it. You should go to share now.
All right. Yes, very good. Okay, let's try desktop 2. See what happens. Yes, there it is.
You don't necessarily need this, but it does. I'm talking tomorrow.
That sounds great. All right. All right, I think we're ready to go.
Can you see it on Zoom?
All right. Now that everything's up and running well, if we can. What's the format going to be? Am I just going to pop right up and start going, or is there going to be like an introduction? I guess that's not your part.
Yeah, Laura.
Okay. Okay, so now can you guys hear me? Sorry. Looks like we were muted. I apologize. All right, so let me start all over again. Thanks for attending. It's going to be the same format as what I did 2 weeks ago, where we want to do some information gathering for this S10 that I'm trying to write to put together to get a spatial transcriptomics machine in the core. So I just want you to, feel free to ask your questions, because, I want to make sure, that we're trying to make sure that we get what's best for the users in the core. So, if you've got any kind of question, feel free to put it in the chat, or, break in and ask questions as you have them. So without that, I don't have much else to say. So I'm going to go ahead and turn it over to Jeff from 10x, and he's going to talk to you about the Xenium system. And I'll just turn it over to you. Okay.
Great. Thank you very much. Thank you for hosting us, and thank you everyone for coming and braving the slippery streets of Atlanta. Appreciate it. I did the same, and I can say it's good we're here together safely. My name is Jeff Byland, and I'm a spatial science and technology advisor at 10x Genomics, which means I'm kind of an in-house technology consultant in the area of spatial biology. And so it's my job from the technical perspective to kind of share how our systems work and give you an idea of where we're coming from in terms of providing tools for scientific discovery. My background is in microscopy as well as omics, and so I've done a lot of image analysis and microscopy over the years, but before that I was doing pharmacology research and doing a lot of omics work there, looking at different cell types. So I'm excited to see these technologies coming together so that we can get images and all of the data that you get from an image, and also the multi-omics, massively multiplex target detection that you can get from looking at the transcriptome and the proteome. So the first thing I'd like to do in this presentation is to get rid of this clutter on the screen. So if you'll excuse me, these are all the Zoom warnings I'm going to get rid of here, hopefully. It's not. Hold on. There we go. Sorry for the folks on Zoom. There's just a lot of Zoom warnings that popped up. Okay. There we go. So hopefully everyone can see now. Looks good. Okay. So at 10x Genomics, our goal is to build tools that allow us to sample biological complexity and to understand it. And if you think about what we do in biology, a lot of times what we're doing is we're doing a bit of a reverse engineering approach, right? We take the systems that we're interested in, we break them down into their individual components. We take organs, we break them into tissues, we break them into cells, we look at their molecular components, and we study how these components work. And then we often try to put these back together in the context of the greater structure so that we can understand how all these parts fit together. And if we take something like the human body, we've got 40 trillion cells, somewhere around there in this organism. And in each one of these cells, we have an enormous amount of complexity, right? We have processes occurring within the cell. We have, different organelles, biochemistry, a lot is happening. And then within organs and tissues that are organized that are made up of organized cell populations, there's a lot of heterogeneity amongst those cells. So, there's complexity at multiple levels. And our goal is to try to unravel this complexity, but not too far, right? If we can't put these into the right context, then we're missing some of our understanding. And so for a long time, there's been the world of molecular biology, there's been the world of molecular analysis, omics, and in those worlds, the idea is you get a lot of data, and it's all basically letters, right? Letters and numbers. And then you have the microscopy world, which is actually where I come from, and we've recently coined the term spatial biology, which I find a fascinating development because for me, biology has always been spatial, since Van Loon had invented that microscope all those hundreds of years ago. And so when I talk to my omics colleagues, I often will say, welcome to space, for the spatial folks, what's exciting now is that we can visualize hundreds to thousands of targets in the context of the morphology. And so that's coming at it from the imaging perspective where we used to be able to see, I remember doing in situ hybridization and being able to see one target at a time. And that was with great difficulty. And then we had fluorescence in situ hybridization where we could see 3 or 4 or 5 targets if we had a good system and a good experimental protocol. But now we're looking at hundreds of thousands of targets. And so it's a really great day for being able to identify molecular targets directly in tissue and to put that in a microscopy context. So when it comes to resolving biological complexity, 10x has been doing this for a long time. So for over a decade, we've been offering our Chromium single-cell RNA sequencing platform. And with this platform, what we do is we take tissues, we break them down into their individual cells, and then cell by cell, we analyze the molecular contents so that we can see which cells are expressing what, which cells have which proteins, and which cells have certain chromatin states. There's a lot that we can do on an individual cell basis. And this has really changed the way that a lot of people do research because now we can get a very, we can take something as complex as the molecular components of a cell, and we can make it into a data set that we can interpret and use to better understand our biological systems. But in this case, we're still missing a component, which is the spatial component. So if we want to now start putting these cells back together and seeing how they relate, then we're going to need to add some extra dimensions to this. And that's where our Visium and Xenium platforms come in. And each one of these platforms is designed around a specific set of applications. There's some overlap, and there have been some developments that have kind of increased this overlap over the years. And then very recently, we've come out with our Visium HD product. But primarily, our Visium platform is for unbiased discovery. It's a whole transcriptome approach where we take a piece of tissue and we can survey the entire transcriptome in that tissue and see where each transcript is located. And we do this by adding barcodes to each transcript that are associated with an XY coordinate on the tissue. And then when we sequence those, then we can locate those in the tissue. And this won't be the primary focus of the talk today, but I wanted to just compare these. It's sequencing based, so it's really a prep assay ahead of going into like in a next-gen sequencing experiment. And we're doing it at the single cell scale. So what that means is that we're looking at the micron level and we're assigning cells and transcripts to different multi-micron areas on an array and using that to what I just got a request to unmute. Are we good? Okay. So with the Xenium platform, what we have is a completely turnkey system. It's all self-contained, and actually the scale here is not right. So the Visium and the Xenium, they look the same in this slide, but actually the Visium is relatively small. It's about the size of a small sandwich press. It operates in a similar fashion. The lid opens and closes. The Xenium is about the size of a benchtop flow cytometer. And it contains here in this portion right here, an automated liquid handling platform. And then behind here is a super resolution imaging system. And the way that it works is we label transcripts here on slides, and then we move those into the imaging system, and then we detect the transcripts that have been labeled. And I'll get into how we do that in a couple of slides. But this system is designed to directly detect transcripts in the tissues, and we're looking at hundreds to thousands of transcripts. So this is not designed to be a whole transcriptome approach at this point. And it's, like I said, high resolution imaging based, and I would say super res because we're getting below 50 nanometers in our ability to resolve where the transcripts are located. And then we are able then to assign transcripts to cells during the assay. And so at the end of the run, the data that you get will have all of the cells that were detected, where those cells were located, and which transcripts are located in which cells. So this is something that shortens the time to usable data when compared to the techniques like Visium that require sequencing after the assay is completed. So this is an overview of our spatial platforms, just to give you an idea of where we're at. We recently released pre-order for our Visium HD, which will begin shipping in a couple of months, which has increased the resolution. So now we can get at a very high resolution the whole transcriptome. So if you're interested in that, please let us know, and we're happy to discuss that further. But we're going to focus today primarily on our Xenium in situ system. And the general idea with this platform is that we're labeling transcripts and then detecting them in the tissue using fluorescent probes that are attached to a DNA probe. And what I'd like to do first is just to introduce you to the type of data that we can collect with this platform to give you an idea of what it looks like. So this is an example of a breast cancer tissue that was sectioned and placed onto one of our Xenium slides. It's about 10 by 22 millimeters worth of tissue. And you can see that we have some really nice morphology here. And what you're looking at is not a stain. This is a pseudo color identifying all the different cell types that were detected during the run. And so what you're looking at then are each one of those colors represents a cell type that was identified and then labeled with a color. And this then gives us a really nice look on the whole tissue level of the gross morphology that we're seeing here. And we can already identify important regions in that tissue on the tissue level, for example, these ductal regions here. And overall, we're assaying close to 710,000 cells and about 110 million transcripts in this one piece of tissue. So already you can see this is a very high content data set that we're generating across a piece of tissue. So at the tissue level, we get a lot of nice detail. We can make out important details. But if we start to now zoom in and we look a little closer at the cellular level, then we can see some additional detail. So amongst all these cell types, you can then select which ones you'd like to activate, depending on what your focus is. So here what we've done is we've identified the endothelial cells with this yellow color, and then we've identified the tumor tissue with this red here. And so what we're looking at is vascularized tumor. And then in another area of the tissue here, what we're looking at is, again, the endothelial cells in yellow marking the vasculature, and then the tumor in this pinkish red. But now we've marked the immune cells with blue. And so we can see in this area the infiltrating immune cells in the tumor tissue around the vasculature. So really nice cellular detail and tissue detail. But we can add other dimensions of data to this assay. One of the things that's unique with this platform is that we're able to preserve the integrity of the tissue throughout the process. So at the end of the assay, you can take the tissue out of the system, and then you can do post hoc assays on that same tissue. This includes traditional assays like H&E, which is shown here. So here what I've done is I have an image of an H&E stained tissue. It's that same tissue that I just showed you that came out of the Xenium assay. And now we've imported that back into the software, and then we can overlay on top of that H&E image the cellular data that I was showing before. So now from the same tissue, we get this level of cellular information, and we can also get a traditional morphology stain like H&E. Now we can also do immunofluorescence, multiplexed IF. We can do IHC. You can do even the Visium assay on a tissue after it comes out of the Xenium run. And so then you can get the context of the whole transcriptome around the in situ data that you're getting here. So if we then zoom in again on this tissue, we can see that the cellular information lines up nicely with the morphology. But as we go deeper, then we get an appreciation for another level of information that we can get from this system. And that's in this final image here. So in this image, what we're looking at are individual cells that have been identified here. So you can see the borders. And then we can see the cytoplasm here. And each one of these symbols that are different colors, it represents a transcript that's been localized within the cell. And so we have, with a high degree of repeatability, the ability to detect transcripts on the subcellular level down to somewhere around 30 to 50 nanometers. And so now we're getting data on this very high resolution level. And not only can we do this, and we can see all these transcripts, this view is a little bit of a mess. I think it's a beautiful mess, but it's a mess because it's all the transcripts that were detected. But we can narrow this down and we can start to identify cells and transcripts of interest using our Explorer software. Here we're looking at immune markers, CD45, 3, and 8A. And you can begin to appreciate how this is almost like a choose your own adventure in situ, where you pick the genes you'd like to see and it will show you those genes that were detected. Now, if we think about this in another context, here we're looking at a mouse pup. So again, this is our mouse multi-tissue phenotyping panel that we're using. And we fit pretty much the entire mouse pup on the slide. And you can see that we're able to detect the right organs in the right place. So pick your favorite organ, you can see it here. We can see that the right cells are in the right place. And we get a really beautiful map of where all these cells are based on the transcript content of each of those cells. And so again, what we're looking at here is not a stain of any kind. This is pseudo-colored based on the cellular identification that we were able to come up with looking at the transcriptome. And of course, on the macro level, this is impressive. But again, often what we're trying to figure out is what's happening on a more fine scale. So if we pick something like the ear that contains a lot of fine features that are involved in sensory perception and balance, then we can, again, get some really highly detailed information in the context of the structure of the ear by looking at markers that are included here. So we can see things like the hair cells, very fine structures like these membranes here, this Resner's membrane that's being identified, and spiral ligaments that we can see here. This system has a really powerful range going from the molecular all the way up to the tissue level that we can take advantage of. And in fact, it's a bit like having a molecular microscope at your disposal for identifying these features in the spatial context. So I hope seeing this data has gotten you kind of thinking about how you might be able to apply this in your research and kind of appreciate why this technology is so compelling. So now I'd like to introduce you to the system itself. And the first part of that introduction will be just to kind of go over generally how we designed the system and what we were thinking about here when we did it. And then I'll go through kind of an overview. Yeah. Great. Yes. So everything that you're seeing here is the same section, same sample, same slide throughout the process. Yeah. Yep. After I give this overview, and by the way, yeah, feel free. So thank you for asking the question from online. Feel free to interrupt. This is something where I think it's very important to have everybody's feedback. So don't worry, this is not meant to be a lecture so much as just, hoping to stimulate some discussion. So when we were designing Xenium, we wanted to make sure that we designed something that was highly sensitive and specific. And when I say sensitive, what I mean is able to detect a broad range of expression from low to high expressors. So we have a really good dynamic range in this platform. And also, when I say specific, I mean two things. So one is that we're able to detect the right transcripts in the right place, the right cells in the right place, but also that we're binding to the proper targets. And this is critical in an assay like this, because this is a fluorescence-based detection approach. If we are not reliably labeling the proper targets, then we can get a lot of confounding false positive signal, and that false positive signal can then become a big impediment to really being able to precisely interpret the data. And this is something that this system has some safeguards built in that prevent us from seeing a lot of false positive signal. It's customizable but biologically relevant, and what we mean by that is that we use panels of probes to detect the transcripts of interest. And we have built quite a few in-house pre-made panels that focus on different organ systems. And I'll share those with you in a bit. But with any of those, you can add your own custom targets. And we feel like this is important so that we take a lot of the grunt work out of finding important phenotyping markers for common cells, and then we leave you some room to add the targets of interest that you'd like to add. Or if you'd like to, you can design a completely custom panel with close to 500 targets in it at this point. So if you don't like any of the panels that we've made, or if you do like them, but you need a different species, I've seen corals, I've seen hairless voles, I've seen social insects, then you can make your own if you'd like. And we work with you on that. And actually, you'll send us your targets and we'll design it for you. So we try to include a lot of flexibility when it comes to being able to detect a wide range of transcripts in any species. This system has industry-leading speed and throughput. Now, I come from the world of pathology and high-content imaging, where throughput, high throughput means you're looking at hundreds of slides in a week, at least. This type of approach is not that type of approach. We're not just standing with H&E and then running a digital imaging run. We're looking at millions of targets in each run. So that takes a little bit longer per slide. But for this type of assay, we're able to get through a lot of tissue. And I'll show you what I mean, because I think it's more informative to share some examples. And then we've also made this very easy to use. And I'll show you the workflow in just a minute. But I think you'll find that as you're looking at this workflow, it's very familiar if you've ever done any kind of tissue microscopy in terms of preparing the sections for running in the instrument. And then finally, we've future-proofed this platform. So we're not in the business of selling a new machine every year like Apple is. We are in the business of creating a platform that will accept the roadmap that we have in terms of innovation over the next several years. I think the Chromium was around for probably 6 or 7 years before we came up with an upgrade. And so the system that you're purchasing today is designed purposely to handle the applications that we have coming on our roadmap, and I'll share some of those with you towards the end of this discussion. Okay, so now that we have a general overview, let's dive in a little bit and talk about what it would look like if you were to run an assay. And at each step, I'll go through kind of some of the technical details and talk about what makes the system unique. So this is the general workflow for running a Xenium experiment. And the starting point is going to be sectioning a tissue onto our Xenium slide. The Xenium slide is made of glass. It has the same dimensions as a typical microscopy slide that you would use. And the technique for mounting a tissue onto the slide is exactly the same technique that you would use if you were mounting a tissue on any other microscopy slide. What's unique about this slide are two things. One is that we've optimized the surface chemistry so that we can maximize adhesion of the tissue to the glass. This is important so that, number one, we can accommodate a large variety of tissues with the same protocol without the need for optimization, and they'll stick nicely to the slide, but also because this type of assay necessarily requires multiple rounds of washing, labeling, and imaging, and we want to make sure that we're not getting delamination throughout the process. And we've been able to do that very effectively here by, of course, optimizing the surface chemistry of the slide, but also we use a very gentle liquid handling approach in the system. This is another unique part of the Xenium platform, is that we simulate pipetting, gentle pipetting in our liquid handling, as opposed to using a flow cell that can introduce shearing factors that can disrupt the tissue architecture. Yeah. Sure. So the question is whether the system can work on OCT sections. Yes, absolutely. So when you're sectioning tissue onto the slide, we can use fresh frozen, so tissues that have been frozen and embedded in OCT and then cryosectioned onto the slide. We can use formalin-fixed paraffin-embedded tissues, pretty common clinical embedding process. And so, yeah, cryosections or paraffin sections are good. The other thing that's unique about the Xenium slide is that we've embedded in the glass fiducial marks. And what this does is it makes it so that we can align the image every time we go through a cycle very precisely. We want to minimize confounding effects that can come from things like lateral shift. And if we are imaging at the, if we're trying to localize at the nanometer level, then this becomes incredibly important. And so we do a lot internally on the imaging side to make sure that we're not getting, mismatch frames or any lateral shift, so that we get a seamless integration of our images. Was there another question? Yeah. Oh, yeah, thank you. So we've tested tissues from about 3 microns all the way up to 20 microns thick. We typically recommend, if you're using FFPE tissues, 5 microns thick. That's the standard for FFPE, but plus or minus a little bit is fine. And then for cryosections, typically we're doing 10 micron sections, which is again the standard, but plus or minus is fine. Really, the issue for samples that are thicker is that you start getting cell layers on top of each other, and this becomes a little bit tricky then to determine which transcripts came from which cell at which Z level. And so to try to minimize that effect, it's common practice in any kind of sectioning, because that's just the case in microscopy in general, is to try to get a thin section so that you're just getting one layer of cells for most applications. So this is similar to that. Yeah, there was a question. Oh, yeah, great. Yeah, great question. So when you're coming into the assay, oh, yeah, thanks for reminding me. Absolutely. Yep, that's great. No, this is good. It's a team effort, right? I appreciate it. We got a good team in here. So I would say, so the question was, are there quality control methods that you can use along the way to make sure that your sample is going to perform well, right? And I would say that this assay is very robust in terms of accepting samples and getting quality data from samples that are even somewhat degraded, right? And I'll show you why in a minute when we take a look at kind of the molecular mechanism that we're using. But in general, we want to make sure that 2 things are good before we even start with this assay. And typically what you'll do is you'll take, if you're coming into a new tissue block, is you'll section a piece of tissue, you'll place it onto a normal microscopy slide, you'll throw a stain on there that like an H&E stain or an IF stain, and you just want to make sure that the morphology is good, right? This is by its nature natively an imaging assay, and so if your morphology doesn't look good, then there's no reason to proceed, right? That's the first thing. And then secondly, we're combining that with RNA detection. So we want to make sure that we have RNA present in our sample and that it's somewhat intact, right? We don't want like an alphabet soup situation where you just have nucleotides kind of all scattered through your tissue. We want to have some continuity. And so what we typically recommend is you do, you'll take a few sections, you'll isolate the RNA from those sections, you'll run it on something like a bioanalyzer or a tape station, and you're looking for, at things like the RIN score, the RNA integrity score, and making sure that you have a certain minimum value. There's no hard and fast correlation, but typically we're looking for something at a minimum of around 3 or 4 for your score. And then you could also look at a DV200 score. That's usually more informative when you're looking at things like paraffin-embedded tissues. And in that case, maybe like somewhere around 25 to 30% as a minimum. But this is a very robust assay. We've seen it work on blocks that are, over a decade old, and it works very well. So a few other things that I'll say about these slides. Oh, was there another question? Did I see one? So right now, the way that the assay is designed is for sectioning tissues directly onto a Xenium slide to begin with. So if they've already been placed on a different slide, we don't recommend trying to transfer that tissue. In that case, you could use our Visium assay with Visium HD. That is compatible with samples that have been previously prepared and imaged. So we would recommend going that route. Yeah. Oh, yeah, the question was, could we use, thank you. The question was, could we use the samples that have been prepped previously? So tissues that have been sectioned, placed on a slide, and then stained and then imaged. Could those be used with the Xenium platform? Thank you for the questions. Okay, so while we're talking about the slides, I wanted to give you an idea of what it looks like when you're working with these slides. And this is important because one of the defining characteristics of the system that makes it stand out is that you can get through a lot of tissue in one run. And this is very important because time is valuable, but money is also valuable. And you can maximize both your time and your money when you're using this platform. So these are some examples of configurations that people have used. Typically, we can fit somewhere around 5 square centimeters of tissue in a run. So we're running 2 slides at a time, and each one of those slides can hold about 2.5 centimeters squared of tissue. And these are some common configurations. So on the left, you're seeing tissue that I've already shown you. This is the human breast cancer tissue. And then in the middle, you're looking at coronal mouse brains that have been sectioned and then arrayed with 4 on each slide. This is something I've actually seen in real life. There was a group at NIH recently that did this, and it was pretty impressive. They got a lot of tissue in a run. And then another way to do this is to use biopsies with something like a tissue microarray. And so at the far right there, what you're looking at are hundreds of samples that are being run in a single run. These are just 1 millimeter diameter biopsies that were taken from tissue. And so then what this means is that we get a lot of tissue per run, so that for the cost of 1 run, you're getting a lot of tissue. But also, maximum cycle time, if you were to maximize, coverage and maximum plex, you can get through this tissue with data in hand in about 2 days. So it's just under 50 hours of runtime. Which is something that I think is quite impressive for a machine like this. And it's definitely the best in class. So that means that, not only we're getting through a lot of tissue in a run, but you're also getting your data at the end of that run. And I'll talk a little bit more about that later. But that's really something that's, I think, enabling, especially in a core environment, for this type of a platform. So that means that in a week, you're getting through somewhere around 6 slides, and you're getting close to 340 slides in a year. So, very productive system. It means you don't have to buy multiple to have a decent amount of throughput. The other thing I'll say about prep is that we've designed our protocol to be universally optimized. So what that means is that it's compatible with any tissue type that is commonly used. And even tissue types like bone or skin that typically tend to be difficult to work with, we have some really nice protocols for using these in our platform. And so there isn't a lot of optimization required up front for each sample type that you want to bring in. We've already done that for you. And again, it's really like just prepping a slide for imaging. You'll section it, place it on the slide, and then you'll move through. So that's kind of an introduction to the slide, the Xenium slide, what you can do with it, and how you can prep your tissue. Then the next step that you'll do involves some prep work on the bench And this is done with normal molecular biology tools in a normal wet lab. Essentially, what you're going to be doing is you're going to be permeabilizing. If you're using fresh or formalin-fixed paraffin-embedded tissues, you're going to do the deparaffinization and decross-linking. But at this step, we're hybridizing with our probes. This takes about 2 to 3 hours of hands-on time. And then there's an overnight incubation. And then we do a ligation step. And this is really important, and I'll explain it in a minute. This is another 2 hours of hands-on time, and then it goes into the instrument. So again, this is very similar to what you might do when you're staining and prepping a slide prior to imaging it, but we're incubating with our probes. Yeah. 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 Yeah, so the question is putting tissue samples onto a slide, and how is this done, especially in the case of the array. So there's a couple of ways that people prepare arrays. One is that they will take punch biopsies and they'll arrange them in a block as an array, and then when they section, then that section has the array already made. And that's typically the most straightforward way to do this. This is a common practice for pathology workflows. The other way you could do it is you could try to float each one of those individual biopsies onto the slide. People have done this, not at that level. Usually that's for a smaller array, or in other words, an array that has a smaller number of individuals, but larger tissues, right? And so typically in those cases, it's a paraffin embedded, and we're sectioning and placing them, floating them onto the slide with a water bath using, pretty standard approach. Yeah, thanks for asking. Yeah, that's correct. It's the same Xenium slide that you would use for any other preparation approach. Yeah, that's right. Yep. So, these steps, like I said, they're a lot like prepping for imaging in a normal microscopy workflow, if you're used to working with tissues. And what we do here is we use a very unique chemistry that makes this system highly accurate, and it can also be exploited to do some really interesting and novel detection strategies. So I mentioned earlier that it's really important that we're specific. And the way that we achieve this specificity in this is that we use these padlock probes. And the way these padlock probes are designed, these are DNA probes, and they hybridize to the RNA target with these hybridization domains. Each one, we have a left and a right-hand domain. Each one of these is about 20 base pairs for a total of 40 base pairs. And if we have proper hybridization, which means that the complementary bases are lining up exactly, then we have a ligation event that occurs. And this ligation then completes the probe, and each probe has here a gene-specific barcode that acts as the fluorescent binding domain. And so a fluorophore that's attached to a complementary oligo will bind here, and this is how we detect the location of the transcripts. And if we have ligation and completion of this probe, then we amplify this binding region, and this amplification then leads to a large increase in the number of fluorescence binding sites that are available per probe, and this increases the signal to noise to the point where one of these small probes is enough to detect a transcript. And we do this so that even if we get some off-target binding where you get some, incomplete homology pairing, then we don't see the probe, and so those false positives never show up in the system. So we require this ligation in order for there to be signal. And this is very important. And this approach has been refined during the commercialization process to where this amplification product is very small, it's very tight, but it still provides a lot of real estate for fluorophores to bind to. And so there are a couple of upshots. One is, of course, it's very specific, right? So our specificity is high. Another one is that we have a small probe that's hitting only a 40 base pair region, and that means that even in degraded samples, our chances of finding the target are high. And this is because all we need is those 40 base pairs to be intact in order to detect, right? We don't need a really long intact transcript to effectively detect our targets. So this makes it suitable and very robust for a wide variety of quality, sample quality. Another point here that I'll make is that we can exploit this ligation approach so that we can do more than just detect transcripts, see where they're located, generally quantify the number of transcripts that are present. We can actually start to generate transcripts that are very accurate and precise, so that run to run they're doing the same thing, and they behave in a very predictable fashion. And that's by generating probes that span things like splice junctions, so that we can determine isoforms of specific transcripts. So not only are we able to detect transcripts in general, but we can get a level of transcriptomic resolution now, and we can start to discriminate between different forms of those transcripts. So this is an example that serves as a very nice validation approach, where we're looking at SNAP25. This is a transcript that is very well characterized as being differentially expressed in the brain. And that differential expression is spatially restricted. And so we can see that we're very nicely able to pick out different isoforms. A couple of examples shown here are SNAP 25, 201, and 202. And we can pick those out and with a high degree of accuracy, and see which cells are expressing one versus the other. Yep. This is something that, in terms of proof of concept is nice, but it's also being done by customers in the field. This was one of the early ones done at the Molecular Genomics Core at Duke University. This is run by Dr. Gregory. We were just there actually talking about this, doing a seminar there. And what they're looking at is glioma, and they're looking at detecting the EGFR splice variants, and how those are affecting the outcomes in the glioblastoma. And what they were able to do is generate some nice probes. So they actually sent us the target sequences, and then we generated custom probes that they could use to identify different variants, and then spatially identify where those are located in the context of the glioblastoma, so that they could better understand how differential expression of this receptor contributed to outcomes in glioblastoma treatment. Now, this is one level of transcriptomic resolution that you can get with this platform that's unique to the Xenium system, again, because we're exploiting this ligation strategy that we're using. But we can also make the ligation dependent on the presence of a single base pair, and we can get down to a single nucleotide variant. And this is exciting because we can now develop probes that detect things like point mutations. So here's an example where we're looking at KRAS, and KRAS, of course, is an important oncogene, and there's the wild type, and then there's the oncogenic mutant. And we can design probes that can detect these based on the presence or absence of this mutation by, again, exploiting this ligation approach and making the ligation dependent on the presence of one or the other in specific probes. And we can do this very effectively, and we can tell which cells are the wild type cells and which are the mutants, and we can detect this with a high degree of confidence across an entire field, and we can quantify these. So again, very important, unique feature of this platform in that we're able to get a high degree of molecular resolution. So, this is what's happening with our probes and some of the things that we can do with these probes. When it comes to detecting transcripts, then we provide a panel menu of different groups of probes that are designed to work with different tissue types, for example. And so this is our probe menu right now. We have these panels that are designed around certain tissue types or that are designed to be whole species. So we have our human whole tissue, our mouse whole tissue, and then we have individual colon, brain, skin, lung, and breast panels. So each of these is designed primarily to contain common phenotyping markers, and then to each of these you can add up to 100 custom genes. And so this gives you a nice balance between pre-validated, ready-to-go kits and the flexibility of adding your own custom targets. And I think there was a question from Zoom. How do we define cell boundaries is the question. That's a good question, and I'll explain that in a couple of slides. So stay tuned. Thank you for asking. So right now, we offer these panels, and then if you see something that you like, but maybe you want a different species, or maybe you want to do something a little different, then we can create custom panels for any species, any targets, synthetic RNA. We have people doing, working on delivering therapeutic RNA that are designing probes against those. And so you can design probes against just about anything that you know the sequence for. We can make those probes. And this is a design process that we work with you on. So we have an in silico prediction algorithm that will optimize the probe design process. It's highly correlative with empirical outcomes. So we've done a lot of work on the R&D side to make sure that when we predict success for a certain probe, that it matches the actual outcome empirically by going back and forth and training this algorithm to the point where it's highly accurate. And so we work with you on this. You'll submit the targets of interest, and then we'll develop a panel. And then we have a pretty fast turnaround time. So from start to finish, which means having custom probes in hand, is about 4 to 6 weeks. And you can even get started today if you wanted to. We have a panel designer on our website where you can log in and create an account and upload your targets of interest and start that design process. We have a lot of experience doing this in just the human alone. We're just over, now we're over 8,000 unique genes. This was several months ago. So we have a lot of experience generating custom content, and we're fairly confident in the quality of that content. So the final step is to place the slides into the instrument and then start the run. And what's happening inside the instrument is that in any one round, we're labeling the transcripts with fluorescent probes of the 5 colors of microscopy that you're typically working with. And then we're building an optical barcode for each one of our targets. So in any one imaging round, we're not just looking at 4 or 5 targets with those colors. What we're doing is we're labeling a number, dozens of targets, and they might share in a given round the same color. Most of them will be dark. And then in the next round, then the colors will be distributed in a different way. And so for each individual target, you'll have a sequence of on-off, and every time it's on, it'll be a different color. And then we will decode this for each one of our targets as the run is proceeding so that we know which target was where. We also take advantage of dark cycles here so that we can more readily locate our transcripts. This means that if a certain transcript shares the same pixel as another, then they're not going to be on at the same time. And so we know during a given cycle which transcript is supposed to be on in which pixel, and this helps us better localize. This is a technique borrowed from super res microscopy that we deploy here that makes it very robust in identifying transcripts. I think there was a question. Yes. So the question is, has there been any work looking at microbial transcripts? And the answer is yes. So there are a number of groups that I've worked with that are looking at the microbiome, or they're looking at host pathogen interactions, both for viruses and for bacteria. So this is absolutely something we can do. Yeah. Okay, so as the system progresses, what we're doing is we're imaging, so we're collecting imaging data, and we're also doing the decoding, the segmentation, the cell assignment, and the clustering. This is all happening on the instrument as we proceed with the assay. And so at the end of the run, no additional analysis steps are required. There's no extra time for waiting for this thing to complete. This is all happening on board, and it's built into the platform. Once it's finished, then it will push the data packet off of the Xenium system onto an attached workstation, and it's there ready for you to do with as you will. So this is, again, very similar to what you're used to with most microscopy systems, where the data, as soon as the run is finished, are ready for you to pick up and use. You can push them from there to your own cloud solution. You can push them to an internal data structure, but it's all ready to go. And we export the data in open file formats, so there's nothing proprietary here. and they're designed to work with commonly available tools, your favorite R-based and Python-based tools. So we export TIFFs, JSONs, ZARs, Parquet files, anything that you're used to working with. And they're also compatible with our single cell data outputs. So you can do things like anchoring with single cell data and integrate these data sets very easily between the two. We also offer an explorer, again, something like what you're used to with your microscopy approaches, where this is something you can download from our website. It's very powerful. There's no additional charge, and it allows you to browse and curate the data in very powerful and easy ways. So you can get started with image analysis right away. Now, yeah. Exactly, that's right. Yeah, one of many. And it comes in multiple formats too. Yeah, that's right. Yes. Oh yeah, the question was, are one of the data outputs a gene cell expression matrix? And the answer was yes. So we've gone through analysis, we've gone through prep, we've gone through running it, we've gone through analysis. I did mention that we can run additional assays on the same section. This looks very good. We've done H&E staining, we've done IF, we've done even Visium analysis on tissues after a run. And we have a technical note that summarizes all of our guidance and learning in doing this so that you can do this if you'd like. We found that it's a very robust thing, very robust approach for both IF and even for Visium. So here we're looking at, on the left, Xenium data, and then from the same tissue, Visium data that was collected. Same section, taken out of the system and run on Visium, and we can see we get a really nice agreement. And so we get kind of like a low-res, high-plex view here, and then we can correlate that with a high-res, more focused data set here. Okay, so I'm going to finish by talking about what's coming up. So one of the questions was about segmentation. Segmentation is very important because this is how we identify how many cells we have, where those cells are, and where the boundaries are for identifying the cells that we bring up in that matrix that was brought up before. And so we are working to put out an industry-leading segmentation solution. Already, we had the best-in-class detection of where the cells are located based on the DAPI stain. So we have a nuclear stain that we include. And so we're able to identify accurately where the cells are and how many cells we have. And now we're adding to this cocktails of membrane and cytoplasm markers that allow us to identify where those cell boundaries are empirically with a stain. And so these will be shipping very soon, so that now in line, you can see the membrane, you can see the cytoplasm, you can see the nucleus, and then we use these for segmentation. Now, we take a very open approach to segmentation. So we will segment, and it's done very accurately. You can look at our data sets, and I think you'll be pleased with what you see over some of the other systems that are available out there. I can, as an imaging expert, I can say unreservedly that this is one of the best imaging and segmentation algorithms I've ever seen. Easily, performs better than the competition. You can buy different cocktails to stain different parts of the cell, and I think you'll find it's highly accurate. Another thing that's coming up is the 5,000 plex panel. So this will be coming in a couple of months. We'll begin shipping this. And so this is increasing the number of targets that we can see by about 10-fold. But because of the nonlinear relationship between the number of cycles and the number of targets that we're hitting, this will increase the time that it takes to run the assay by about 2.5-fold. So we're getting a 10-fold, an order of magnitude increase for only about a 2.5-fold increase in time. So we're looking at about a 5-day machine runtime to get up to the 5,000 plex, which again is very fast turnaround time for this type of assay. We're also going to be adding inline protein multiplexing. So this is coming towards the third quarter of this year. We'll be releasing antibody panels that you can add to the run so that you can do RNA and protein detection in the same run in the same tissue. And so we're excited about this because now we're looking at both the proteome and the transcriptome in the same section at the same time in the same instrument. And so, you know, you will have all those layers of information. And then by the end of the year, we'll have kind of filled out our offering in terms of panels. So you can go all the way from 1 up to 5,000 and everything in between. So we'll be adding, other 1,000-plex panels that kind of match what we've already done with our other panels. So we want to continue to offer flexibility and compatibility with this platform. So some exciting things ahead, and these have been slated, and stay tuned for more announcements to come. I'll finish by just recommending that you go ahead and take a look at our data sets. We've got close to 20 data sets available online that you can pull down and you can take a look and you can see. I think you'll find the quality of the data, both from an imaging perspective, but also from the transcript detection perspective, speaks very highly of how this system stands out. And I'll go ahead and stop there. So thank you for your attention today. I think there may have been another question. Okay, great.
Great information. So everybody online, especially this is recorded, so we're gonna put it online. So if you missed anything and folks had to leave early or came in late, we're gonna put this on our website. So, and then look for those of you, look for a follow-up email from ICI about this. We want to do some follow-up input on this seminar as well as the other one. So we'll be reaching out to you in the next week or two about that as well. So if we don't have any more questions, we'll just end this and say thank you guys for your attention and coming to this, and we really appreciate your input. So thank you.
Thank you very much. Yeah, it's really important. So one of the things that we can do right now